Ryan, welcome.
Thanks.
So I believe Ryan comes here today from Oxford. He's the CEO of Oxford Genetics. Now, I know you started off in Oxford University.
Well, Leeds actually.
Leeds,
Then on to sunny Oxford.
Yeah, indeed. So you started off, I believe, as a traditional academic. So how did you make the transition? What made you decide to start the company?
When I did the PhD, I had the benefit that my PhD supervisor was incredibly flexible. He sort of let me take my research in my own direction. So you get to the end of a PhD and then you're looking at doing potentially a postdoc, and what you rapidly appreciate is not every principal investigator does exactly that. So you sort of look at them and you think, actually, I'm going to have to be forced to research in this sort of area or going this direction. And the company, or starting a company, at least had the vision of taking it in my own direction. So having being in charge of my own research and pushing it in my own direction. Obviously had the downside that you didn't have any money and you were trying to do it on a shoestring, but simultaneously it gave you the freedom to take your research where you wanted to go.
So the name, that's an interesting name. Oxford Genetics.
Yeah, well, it sort of does what it says on the tin.
Yeah.
So, yeah, I mean, as time's gone by, I've appreciated that calling it Oxford Genetics was maybe not the wisest thing in the world. I often get mail from Oxford Biomedica or Oxford Gene Technology or various Oxford other companies. So it's, yeah, I mean, it still does what it says on the tin. But yeah, I mean, we considered changing it to Oxgene, and actually all of our domain names are Oxgene, and we considered it in the early days, but yeah, just stuck with it for simplicity, really.
So Oxford Genetics does three different main areas, I believe. So gene therapy, gene editing, and antibodies. So these are two very hot areas right now. Has that popularity contributed to your company's success, do you think?
I mean, the founding principle of the company was sort of more a sort of synthetic biology type approach, and that was doing molecular biology is very complicated, and DNA is inherently very complicated. So what you ideally would like to do is start by standardizing the blocks with which you're working with, confirm that they work in a certain way in a certain organism, and then use that block consistently in that configuration with other parts. So sort of part A times part B times part C gives you a defined output. The company was founded with that principle, and what we did was develop around 20,000 different pieces of DNA, which we then characterized in different organisms. And really what happened was we were going out to market, and people were no longer interested so much in bacteria, they were no longer interested so much in yeast, they were no longer interested so much in phage, but what we kept getting asked about was doing work from companies working in mammalian cells. And so then we sort of pushed the company in that direction after about 18 months, and then again, what kept happening was I was going out talking to clients about all the great pieces of DNA that we'd validated, and actually they were only fundamentally interested in three different things. We were talking with companies who were developing gene therapy products. Their main pain point was largely manufacture. We were talking to companies who were trying to discover antibodies against difficult targets, like things like GPCRs, and so we started to develop technologies around that. And then gene editing, and really the main challenge we were finding in gene editing was people wanted scale. They wanted to be able to edit 100 cell lines simultaneously. You know, they wanted to take a gene and knock every single amino acid out within that gene and do it at a price point and a quality that made sense. And so that's really where we've ended up pushing in that direction. But it's kind of, you know, if you have a lot of pieces of DNA that you validated, you can go after largely any market, if that makes sense, if you're working in biology. And what's happened is we've ended up going into the three markets that the industry has come back and told us they've got an issue with, which is largely drives our R&D.
So a lot of biotechs, I say, don't necessarily focus on the sort of large-scale automation side of things. I mean, but I think it's really quite an important area, particularly with things like, as you say, with the gene editing and I mean, why do you think that is? I mean, what's your sort of unique?
Well, I think the problem you often have in a biotech is that it doesn't think like Ford would if they were making cars. You know, we scale an inefficient process. You know, you have a technician doing a process, maybe it's an ELISA or something like that. You have more market demand for more projects coming in, and what you do is, my solution is I'll buy 3 more technicians. That all of a sudden means you've massively inflated the bottom of the business, you know, with technicians, and no offense, but humans are generally quite error prone. So, you know, at some point you start to get inefficiencies in that process and weak links, and the whole thing starts to fall to pieces on quality, which is ultimately why the car industry is based entirely on an assembly line-like process. So for CRISPR, when we realized we were going to be trying to make 1,000 clonal cell lines next year, we're not going to be able to do that with just technicians. And plus you'd have a lot of very unhappy technicians doing the same thing over and over again. So what we did was pretty much automate the process from end to end. Yeah. That has the added benefit that you then have consistency and quality throughout the whole process. So it was largely driven by customer need and demand, to be honest.
Okay. It's interesting. So you're obviously doing something right, as you started in 2011, and I believe you now have almost 90 people, is that right, working for you?
Yeah, it's about that.
So how did you go about getting things off the ground? I mean, any tips for anyone here who might be thinking of starting their own company?
Well, I mean, I wouldn't start it the way I did. I basically finished the PhD, had no money. I agreed the PI that I'd done my PhD with to put some, I convinced him whilst driving a car and on a shake of a hand to put some money into the company. I then took out a bank loan because I didn't have any other way of getting any more money to start the company. And then I went round and met with about 20 different biotechs in the Oxfordshire area and said, do any of you need molecular biology services, just making pieces of DNA, because I can do that. And there was one that I found that was willing to give me a bench in exchange for me making a series of pieces of DNA for them in the weekends and the evenings. And so all of a sudden I had access to this great laboratory for free rent, still had very little money, but it allowed me to get to the point where I built a website, started to get some sales, and basically shoestrung it, to the first 2 or 3 years. It was pretty painful just doing that. I think with hindsight though, I don't think I had any other choice, because who's going to trust you when you just come out of a PhD, you've got no commercial experience, you're trying to convince people that you can do something that you've never done before. I mean, you'd be nuts if you did invest, to be honest. So it's kind of, with hindsight, you have to prove yourself and get it to a point where you've gone from an idea to some sort of value inflection point where an investor or somebody else can come in and say, yes, okay, this actually now has value. So I don't think I really had a huge amount of alternative. But the thing I would probably say if somebody's going to start a company is that you will convince yourself that it's the right business model so much easier than anybody else. So you will tell yourself this is the right thing to do, and there is a market there, and there's going to be thousands of customers. It's going to be really easy to scale. It's going to be really easy to sell. And after a while, what you realize is maybe you're wrong. Maybe the market isn't quite how you thought it is. Maybe the customer doesn't quite want what you've been developing for the last 6 months. And so then it, from my perspective as a biologist, always comes back to something that Charles Darwin quite pointedly pointed out. It's not the fittest or the most intelligent, it's that which is willing to adapt to change that will survive. And so as somebody running a business, you often have to look at what you've made and go, okay, actually, can I use this in a different way? Can I service a different market? Is there a way to make more value from it? So, I'd say that's it's a rabbit warren that you can easily run down as a founder of a company.
So do you have any examples of, where you might have you went wrong and then you sort of ended up turning it around or anything like that?
Yeah, I mean, so the company, originally, the model was make all these pieces of DNA, put them in the catalogue, sell them on the website, which is fine. We did that, and we still make good money from it. But there's relatively limited market. As in the number of people that are genetically engineering that are out there is quite significant. There's a lot of people doing it. Sounds like they're doing it next door. But actually, the number of people that are actively looking for the piece of DNA that you've got in your catalogue at any given time is quite small. So, in the end, the market that you can actually get to is relatively modest. But if you can start to use that to develop new technologies, which you can license out, or you can use to modify cells, which then turns a £200 sale into a £20,000 engineering project, then for us that was a pivot to trying to see, saying, look, okay, this is fine, the catalogue can stay there, but actually we need to figure out how we use the catalogue for something more lucrative and ideally build shareholder value through things like licensing or partnerships. So yeah, that was definitely an example of pivoting the company in a certain direction.
Okay, so I mean, I come from a science background, and in my experience at least, there's been, a lot of academics push back against industry, and there's a sort of, you're going over to the dark side thing. I mean, I'm just interested to hear, did you get any of that, or? Yeah. So perhaps you could say a bit more.
Yeah, so when I did my PhD Viva, I'd started the company a little bit before, and the very last, after the Viva, 4 hours of being grilled, the external examiner said to me, what are you going to do next? Exciting career as a scientist in the university. I said, no, I'll start a company, I'm going to do that. And he said, I've just got one piece of advice for you. You'll wake up in 5 years and you'll be miserable. And I didn't really know what to say. Like, you know what I mean? I just thought, fine, I'll risk that because I know I don't want to do a 2 to 3 year postdoc cycle personally, and there's nothing wrong with that. But I didn't want to do that personally, and I knew I would be fundamentally unhappy doing that. So, but I was quite amazed at that level of vehemence against industry. I think it's changed. I think it's changed a lot. A lot of it's changed because of there's a lot more spin outs. A lot of scientists are now looking at how they apply their science commercially. I mean, if you look at Oxford with OSI, this investment portfolio that they have within Oxford, the number of spin-outs, I mean, there must be 60 spin-outs in the last 5 years. That changes the whole dynamics of the way the university thinks and the way people apply their research. So it's definitely changed, and it's changed for the better. But I think there's still a lot of people out there that think that the commercial world is the dark side, despite the fact that 90% of their research budget is buying reagents and materials from commercial companies.
Well, quite. Someone's got to fund it, right?
Yeah, exactly.
I mean, so you went from a very small scale to what's obviously now reasonably large scale, or certainly a lot larger than it was. I mean, you must have had to go through a lot of changes during that time in terms of what you personally were doing in terms of work. Like, I wonder if you could talk a bit about that. Yeah,
I mean, in the early days, I mean, you're literally doing everything. You're like doing the accounts, doing the web maintenance, making the products, shipping the products, sweeping the floor. You're doing literally everything. And then at some point, you start to get somebody else to start to do these different tasks, and you have to sort of assess what your role is in the company. And I say I've had to do that probably about 4 times over the last 7 years, that the job has completely fundamentally changed on a daily basis from what it was to what it is now. My job largely now is, if I'm doing anything operational, I'm probably not doing my job correctly. I should be doing everything that's strategic, which is looking at what our competitors are doing, what we've developed and how we can apply that to the market, looking at how the company is structured, how it runs strategically. And so I don't get involved in the sort of nitty-gritty of how many plasmids we've sent out or how many cell lines we've made and things like that, because as soon as I go into that, I'm no longer keeping my eye on the bigger picture of where the company sits within the world. So that's a recent transition for me. I was still involved. We didn't even have a CFO until about a year ago, so I was still doing a lot of the finances and the accounts back then. And as soon as that CFO started, about 60% of my workload changed. I didn't have to do that. And I sort of sat there and I thought, this is strange. I don't actually know what I'm supposed to do today, because I was supposed to be looking at the spreadsheet, and today I'm not. So yeah, I mean, that was quite a revelation and quite a happy one, I have to say. But yes, it's changed a lot.
So is that something you enjoy? I mean, have you enjoyed the transition, or do you miss being back in the lab?
I mean, lab work is basically 90% failure. So, I mean, literally it is. It's the most depressing thing in the world. You do the same Western blot over and over and over again, and then you're just changing the dilution of the bloody antibody. And then for some reason, that day it works. Going from 1,000 to 2,000-fold dilution, that was the difference. And I mean, it's just, I mean, I quite like lab work, but I think the fundamental reason I started a company making DNA is because cloning pretty much always works. so I don't miss the failure.
Famous last words.
Yeah. I mean, by and large, I don't miss the lab work. I don't miss the failure of the lab work, but I miss being able to get into a specific scientific problem and figure out the logical next step based on the data I've got. I do miss that. That's where you get exciting science. You get a result that you weren't expecting, and it sends you off in a direction that is different to the Gantt chart that you'd built on the assumption that the data was going to be that way. So I do miss that, but yeah, I mean, it's a different job, and I made my bed. I have to have to lie in it.
Okay, yeah. So I wonder, what's your business model? I mean, how did you go about attracting investment once you got onto a slightly larger scale?
Yeah, I mean, I think the business model itself, what we try to do is everything that we do, we want some kind of downstream revenue or mechanics that is beyond the project that we're doing for the client. By that, what I mean is, you do a project for a customer, they take it back into their manufacturing facility, or they take it onto clinical trial. There's some type of license that is at the back of that specific project that means that you get longer term value or shareholder value, which is ultimately what it's all about building within the company, from the project that you've done. There's 2 ways in which we do that. One is that we will develop something which can add to our catalogue. So maybe it's a piece of DNA, or maybe it's a cell line that we've edited. We can add that into the catalogue, so we can hopefully get some future sales from that as well. Or we do a service and we develop a technology which is either licensed exclusively to the customer, or it develops a platform technology which we can license across the industry to 50 to 100 other companies. So that's the way in which we try to build the business. And what it means is a lot of these licenses have repeat revenue year on year. And so you can start to see over years after year, these things start to accumulate. And so it starts to make next year's revenue look a little bit easier to get to than having to find every project every year twice over, because you're trying to grow the business.
So you mentioned that you sort of started out with the synthetic biology side of things. So obviously that's been a really developing field over the last few years. So I wonder, what are the sort of hottest things you think in 2018 in the synthetic biology side of things?
It's just a crazy, crazy time to be in biology and biotech. I mean, it's just nuts. You know, it's like, if you think about the linearity of medicine and what we've developed over the last 300 years, the windows of cycles of innovation have got more and more compact. You know, you've got sort of 300 years ago vaccines, and then there was 200 years until we discovered sterile technique, and then another 50 years until antibiotics, and another 10 years until chemotherapy, another 20 years until antibodies. And now you've got regenerative medicine, stem cell medicine, chemotherapy, CAR T, gene therapy, antibodies. I mean, it's just crazy the amount of stuff that's being developed so quickly. I mean, I think CRISPR is obviously a very exciting area, and also a lot of controversy. But
recent news.
Yeah.
But so you'd have to pick gene editing then, if that was would that be your top pick?
I mean, there's the two I would pick are gene editing and gene therapy. Yeah. What's really exciting is you can combine the two. Yeah. Right. So you can put the gene editing apparatus into a gene therapy vehicle, a viral vector, and do gene editing using that. So you're basically combining the two coolest things right now, and that's a lot of what these companies are doing, either ex vivo using lentiviral vectors or in vivo using AAV vectors. But I think that's a really exciting combination of two different technologies.
So do you think, obviously, as I understand it, there's been a real revival of gene therapy with recent successes such as the the eye therapies and various things. Do you think that's really helped in terms of your business? Obviously, I know a lot of people have said that there's trouble finding enough of the vectors and things like that, or having supply chain issues.
My PhD was about half of it was in gene therapy. The company has kind of found itself back in that direction largely by market demand, and the real issue that we try to solve is manufacturing enough of these viral particles. You know, some of these viral particles give you like a mild cold, and we're trying to treat patients with 1 × 10⁻¹8 or 16 viral particles or whatever it might be. I mean, that's crazy amounts of viruses that you need to manufacture, and what it means is at the moment the cost of goods is something like $1 million per treatment, for something like haemophilia. When you then want to apply these really cool technologies to something that affects a wider part of the population, like sickle cell or something like that, you can't pay £1 million per treatment for that, and so it's largely down to the manufacturing process. And so what we try to do is make that more scalable. It's basically, we're trying to transition the industry from being a very batch-driven process where you take bits of DNA, you put them into cells, and you make some viruses in a small batch, to a very scalable bioreactor suspension type process, which is exactly where antibodies are right now, but it's not where antibodies were in the late 80s when the first drugs started to come to market. So the gene therapy industry is going through that exact same transition as the antibody industry went through, and it took about 5 to 10 years for that process to happen, and we're hoping to push the industry in that direction.
So I imagine, I can see how that would be very attractive, because if you're managing to reduce the pricing, because I know things like Luxturna, for example, I know that's like $800,000 or something per patient. So I think I'm sure there would be a lot of people who would be very happy if that price could come down.
Yeah, I think it's one of those things where we want gene therapy to be successful, and if we think about it clearly works. There's plenty of examples with haemophilia, with retinal disorders, it clearly works. So what's going to make it not viable? And it's the cost. It's the cost of doing it. People complain about the NHS, but the NHS was founded at a time when we'd only just discovered penicillin. We had a bit of paracetamol and a bit of ibuprofen. You rocked up and got a bandage. Now we're asking the NHS to pay for £100,000 injection of Herceptin or whatever it might be, or £1 million for an AAV treatment. I mean, the costs need to come down. I mean, I think fundamentally the model probably needs to change to the way we're paying for it, but these drugs are just way too expensive to treat a mass disease, and we want it to treat a mass disease. So, and antibodies are in exactly the same problem, in the late 80s, early 90s.
I think this is a very good point to open it up to the audience. Does anyone have any questions?
Yeah, do you think the antibodies have moved faster than gene therapy? I mean, gene therapy went up, plateaued, do you think? So that's now going up again, but antibodies are really booming in terms of therapeutics in the market.
Yeah, I mean, I think antibodies got there slightly earlier, not a lot earlier, but slightly earlier. I think gene therapy had a few bad turns. I think the problem with gene therapy was that you could use any of probably 50 different viruses for gene therapy, and within each of those viruses you could use 50 different serotypes or isotypes or species of those viruses. And actually a lot of it was just going through and finding out which ones worked. Gene therapy had a couple of knockbacks in the late 90s. There was the treatments for ADA SCID, where some of the patients developed leukemia. So that basically took everybody in vector development back 5 to 10 years to try and improve those vectors so they didn't cause leukemia. There was also a patient that died from particle toxicity from another clinical trial, completely separate virus, which again set it back. And fundamentally, there was no clinical success. Largely things weren't working. It was only in about 2011 when the haemophilia trials came out where actually everybody was like, wow, gene therapy is actually really working. And that's when AAV exploded. CAR T as well, similar time, started to work for leukemia or myeloma. And so it was, I think it was a matter of trial and error of a lot of different things over many years. Whereas antibodies, they exist already in us. They do a job. It's a repurposing of a piece of equipment that we know already works. And their circulation in the blood is 56 days, the half-life is. A virus, if you inject a virus intravenously, you're lucky if the half-life is more than a minute. So it's, they're completely different molecules. And I think it was a matter of just trying the different ones and finding the one that was right for the right application. Because lentiviruses don't really get used in the bloodstream. They don't really get used intravenously or injected into any organs, but they are amazing for ex vivo, for CAR T applications. And, adding the virus onto the cells without the bloodstream there, they work fantastically. They integrate into the chromosome, the cells go back in, the cells divide, and so the virus is carried with them. AAV is not the same. So it's completely different tools for completely different applications. And so I think it was just a matter of going through them and finding the ones that were right and worked. So I think, yes, antibodies definitely have. I think the fact that viruses or viral vectors can interact with the cell, and you could even program the virus to respond in a certain way based on the cell. So the cell gets a signal, maybe something becomes tumorigenic around it, and it could actually trigger and produce something that then kills the tumor. So that in terms of concept and what you can do with a viral vector in comparison to an antibody, I think you can do a lot more because you can build in whatever you want, essentially. So I think the potential is huge for viral vectors.
So I think there's a gentleman at the back there.
Hi Ryan, I'm Faith, the science entrepreneur. I just wanted to ask, how do you build your company at an early stage? How do you get people attracted for your company, and how do you spot talent at such an early stage?
Painfully. Yeah, I mean, if you're doing it for the first time, I mean, I am a big sort of fan of just trying to figure it out initially yourself, because I think what I found was that although I don't now want to be involved in how a profit and loss account works and how you recruit the best people. I mean, I'm interested in that, but all of the basics of how you run a company, when you're setting it up, you kind of want to do it yourself, because if you then bring lots of people in and then they understand it and you don't, then it actually makes it more difficult for you to scale the business because you don't understand the problems they're facing. So I hate a lot of the things that I've had to learn about patent law, about recruitment, about HR, about all of these things that I just had to learn as a small company, trying to do it yourself, that fundamentally I'm not doing now, but I know how they all work. So I'd say as much as it's really painful in the early years, it's actually to your benefit to get stuck in and learn it and understand it. But yeah, I mean, I'd probably say for the first couple of years, every couple of days I wanted to quit, but quitting's easy. Staying the course is what's difficult. I can't remember who said it, it's a famous line, but somebody says you just have to hold on to the tiger. And it's basically it. You just have to keep pushing and keep trying. And all of a sudden stuff just starts to come together. It takes ages, but eventually it comes good. So.
Was there a key moment you feel like you really hit really started going well?
I mean, I think when you get a couple of employees who are either better than you at certain things, which is ideally what you always look for, is an employee that's better than you at the thing you're trying to get them to do. That's the moment when you can let go of that piece. You give them something, they do it either quicker than you or better than you or as good as you, ideally better and quicker. But and you sit back and you go, wow, okay, actually, if I'd done that, it would have been less efficient and I would have been distracted from these other things. So I can now give this to that person. And it's a matter of just finding the right pieces in your puzzle that you fundamentally trust with those areas and surrounding yourself with those people, because that allows you to step back and think about the bigger picture. And so that it's sort of incremental steps, and that's what I was saying with the financial side. I found somebody who I could just confidently just give all that to, and I didn't have to think about it so much. So it's about finding the right people.
So how far in was that?
Probably about So it was one person for about 2 years, just me.
Yeah.
And then the first person I found, who's still with the company actually, was back in about 2013.
Okay.
Yeah, molecular biologist, very gifted molecular biologist. He heads up all of our CRISPR library work at the moment. So yeah, and then that followed with a couple of other molecular biologists who, again, the same. One of them now does a huge amount of our viral vector work, still with the company. So yeah, but you can recognise those people because, as I say, they're better than you. So which is weird, but you know, it's great. It's really good.
So, anything else?
So you, as a gene therapy company, gene editing company, and the recent hype around Moderna IPO, what's your views on Moderna approaches, and is it complementary to what you do?
What, as in the combination of gene editing and gene therapy? Yeah, no, I mean, it's an incredibly exciting area. I think it's probably the most exciting area of you know, new therapeutics in terms of the direction that we're going. I think the gene editing space is challenging for a lot of reasons: IP, off-target effects, you know, various concerns about what weird professors are doing in China. You know, it's got lots and lots of issues around it, but as a potential therapy, I think it's amazing. You know, it sort of smashes everything else out of the park in comparison. So.
That's something in general. When you actually look at Cambridge, Massachusetts, or you're looking at...
It's a dirty word. You can't say that to a man who's called his company Oxford Genetics.
Despite Brexit and everything, are you quite optimistic in the fact that, do you think we can get to that stage? Do you think we've got the entrepreneurial... Because we've got the talent to get there. There's no question about that. We've got, you know, but do you think we've got the... In 20, 30 years' time, I know it's looking at the glass ball.
Yeah, I mean, I think we have. I think we've, you know, I have to say, you know, one thing to the government's credit, they have supported UK biotechs quite a lot, particularly. I mean, we've had 5, 6 grants, I think, over the last 7 years, which have supported, you know, a lot of our R&D projects. So I think the infrastructure is here to build it. I don't think there's a problem with that. I think there's maybe a fundamental difference between British people and American people. That, you know, when I go in to talk about a deal with my American head of sales, like I say, I think it's a 200 grand project. He said, okay, we'll start at $1 million. You say, you know, there is a difference to the way in which they work in terms of business to the way we work. And I also think the appetite for risk from investors to a degree is also different. It's not to say you can't find the right investor in the UK, but there's less of them, I would say, that are willing to take that punt very early with large amounts of money in the UK than you would find in the US. But I don't see any reason why not. I mean, OSI and Oxford is a classic example of that. You know, I don't know how many spin-outs we had in Oxford in the preceding 10 years to when OSI was created, but I'm sure we've created 10 more in the last 5 years, you know, 10 times more. So I think you know, it's about getting the right pieces, you know, in the right locations. But no, I don't see any reason why not. I mean, Cambridge, UK is doing pretty well. Yeah.
Okay, anyone else? Gentleman at the back.
You mentioned the word risk. Entrepreneurs, the risk takers by how you are. So how did you manage the risks involved in what you were doing in terms of insurance risk?
In terms of insurance? As in, do you mean actual insurance?
Yeah.
Well,
if something goes wrong, you could be in, particularly in America, you could be in for a lot.
yeah, I mean, yeah, we have the benefits in the UK, obviously not, we don't have so much damages and things like that in the UK as you would in the US. Yeah, exactly. We're not quite so litigious in that regard. But, I mean, I had the benefit when I started the company that I was working in somebody else's labs. So they were largely, they took the risk, but not me. But I mean, again, one of those areas where I just had to sort of find it out as we went. You know, there were certain insurance policies I didn't need until I had one employee, and then all of a sudden I had to figure out what the risk was of doing that. And as we've gone on and we've added things like gene therapy systems and CRISPR-Cas9 systems, you know, you have to implement all sorts of safety policies within the company that you never had to do before. You know, if you're working with just E. coli for molecular biology, it's relatively innocuous. But if you get lentivirus in your eye, you know, that's pretty dangerous. And so, you know, you have to all of a sudden start to assess every single thing that you bring into the company with a completely, you know, new health and safety policy. So we've had to do that a lot. It takes up a lot of time, but you know, when you've got a lot of employees, you really have to worry about that. So.
Okay, gentleman at the back there.
Yeah, so how did you go from having an idea of actually, I think there's a gap in the market, how did you go from that to actually thinking, I'm actually going to go out there, put myself out there and dedicate myself to starting this company?
I don't know really. I'm not quite sure what the day or moment was when I decided to do it. I know that I was working with a level of dogged determination to make it succeed back then that, you know, it scares me, the level of risk that I was willing to take to make it work back then. You know, if you ask me if I would take the same risk now, you know, starting it in the same way, the bank loan, no, the answer is no, I would not. So I think it was just that I really, really wanted to do it, and I really, really wanted to make it work. And I really wanted to do it initially, and still continuing, but in an area that I felt would give me reward. And so developing bits of DNA that I knew other researchers around the world would be using for in their research, I was getting a level of reward from building those pieces of DNA. And so it meant I felt like I was building towards something of value. So it was almost sort of like you had to convince yourself that it was worth doing, as much for yourself, but also because you were pleased with the output of what you were doing. So yeah, I think, but I don't remember a specific moment when I stupidly convinced myself to do it. I'm sure there was one.
Okay. I think one more question and then I think we need to move on, but you can always grab Ryan later. But yes.
We're at a networking event. How do you use networking to promote your company both within the UK and globally?
I don't know whether you know about Charles Darwin, but he had a mirror outside the front of his house so he could see who was coming up the thing, and if he didn't like them, he would run into a back room. So I've generally, in the early years, avoided networking, partly because, to a degree, when the company's really small, you're not as proud of it. You know, you're not embarrassed by it, but when somebody says, "Oh, you're a CEO of a company, oh, how big is it?" and you say 2, it's not, you know, so it's a bit difficult to network confidently when the company's that small. As the company's got bigger, we've got business development people who come into the company who, by their very nature, are just fantastic at networking. You know, again, finding people that are significantly better than you are at doing it. And we've got some people in the company who are just fantastic. They can break down any door you want them to break down. You just give them a name and they'll go and find that person. So I personally don't do a huge amount of it. I do come to events like this and, you know, make an effort, but it's not something I actively do. But the business development team, certainly a lot of our leads come from those activities, you know, going to meetings and just talking to people, and also feeding that back into the R&D. You know, if you go to a networking event and everybody's saying the same thing at this gene therapy conference about X, feeding that back into the company, that sort of drives the R&D as well. So you get a lot of back from it as well as you're pushing it out there.